Welcome back to the College of Glycation. I'm your host, Paul Reynolds. I'm a biomedical scientist and professor of cell biology. Today, we're going to dive deep into a fascinating topic that's building right on what we've covered in previous episodes. In this case, we're talking about the current developments in the analytical methods for advanced glycation end products or AGEs, specifically in foods. If you've been following along, you'll know that we've talked about the basics of glycation, how these sneaky compounds form in our bodies and in the foods that we eat. We also have talked about why they matter for metabolic health. But today we're zeroing in on a comprehensive review paper that was just published by Hiroyuki Kataoka in October, so just this month or last month, 2025. They published their work in the journal Molecules, and that work pulls together the latest trends and challenges in how scientists are detecting and measuring AGEs in everyday meals. So, over the course of this podcast, I'll talk about some technical terms, but hope to bring it back to real life application. The researchers in this case were based in the School of Pharmacy at Shujitsu University in Okayama, Japan. So let's think of their work as the detective work behind understanding just how much of this glycation product is lurking in the foods that we eat, like the steak that's overly grilled, or milk, or even a morning coffee. We'll keep it straightforward, evidence-based, and relevant. And again, although there are some technical terms for specific AGEs and techniques they employed, I'll end with a conclusion statement about how to apply them. There's supposed to be no fluff in this discussion, just the facts to help you make smarter choices about what goes on your plate. And since this is a key publication pulling from over 20 years of research, we'll expand it a bit on its insights and give you a fuller picture. So let's first start with a refresher on the basics of glycation, since this episode assumes you've got the groundwork from our earlier conversations. Glycation is that non-enzymatic reaction where sugars like glucose or fructose stick to proteins or lipids without an enzyme being involved. It's the Maillard reaction in action, the same process that browns your toast or gives overly grilled meat that crust that you're familiar with. In food, it kicks off when reducing sugars react with amino groups on proteins. The first step is it forms a Schiff base that rearranges into what are called Amadori products. From there, things get a bit complex. There is dehydration, oxidation, and rearrangements that lead to reactive dicarbonyls. Some examples include glyoxyl, methylglyoxyl, and 3-deoxyglucosone. These are the bad AGEs that then cross-link with amino acids, specifically lysine and arginine, to create the stable AGEs. In this case, we're talking about compounds like N-epsilon carboxymethyllysine, or its cousin, carboxyl ethyl lysine, purine, and a few others. Some of these are free-floating, low molecular weight versions, but others are bound to proteins or peptides irreversibly. In foods, especially processed or overheated ones, AGEs form fast and in large quantities, way more than what happens slowly in our bodies under normal conditions. As Kataoka details in this review on the formation mechanisms for AGEs. Now, why does this matter? Because when we eat these dietary AGEs, or D-AGEs as I'll often call them, they don't just pass through us harmlessly. Sure, only about 10 to 30% or so are absorbed into the bloodstream, But once they are there, they accumulate in tissues and bind to receptors like RAGE, the receptor for ages, and triggers in the process oxidative stress, inflammation, and cellular dysfunction. Over time, that buildup contributes to a host of issues. For instance, in previous episodes, we've talked about diabetes. Elevated blood sugar ramps up endogenous age formation, exacerbating or worsening complications like diabetic retinopathy, where ages damage retinal blood vessels, or neuropathy, as well as also considered. In nephropathy, stiffening of kidney tissues and impaired filtration results. Now, studies show that ages promote insulin resistance by modifying proteins in insulin signaling pathways, making cells less responsive and worsening hyperglycemia. Beyond diabetes, these ages are linked also to cardiovascular problems. Imagine atherosclerosis, where ages cross-link collagen in artery walls, making them rigid and prone to the buildup of plaque. In the brain, they play a role in neurodegenerative diseases like Alzheimer's disease, accumulating in areas that fuel neuroinflammation. Even in the process of aging itself, ages accelerate skin wrinkling, bone fragility, and cognitive decline by cross-linking extracellular matrix proteins. And let's not forget about emerging connections to gut health, a topic we talked about a few episodes ago. In addition to the gut, liver disease is rampant and other reproductive issues. All of these are where ages can disrupt the microbiome balance, promote fatty liver, but also impair fertility by damaging sensitive tissues. So the evidence is clear. Chronic exposure to high dietary ages or DH levels from processed foods isn't just a metabolic nuisance. It is a low-grade assault on multiple systems, synergizing with the endogenous ages you already make to heightening risks for these conditions. So in this publication that we're talking about today about the age in the diet, Kataoka and colleagues emphasize the linking of food ages to broader health hazards. But here's where Kataoka's review really shines. To tackle these risks, we need precise ways to measure ages in foods. They do not, nor do I advocate your measuring ages in your kitchen, but these techniques, when employed in a laboratory setting, can further characterize foods for the ages they contain and inform the public about better dietary choices. Without good analytics, we're merely guessing at exposure levels. Over the past 20 years, methods have evolved, but challenges remain due to the age's diversity. There are over 40 types that have been identified, and food complexity has gone up. This review breaks it down into sample preparation and detection techniques with a massive table that summarizes dozens of studies on foods from infant formulas to roasted meats. For free ages, extraction uses water miscible solvents like methanol, for example, followed by deproteinization with agents like trichloroacetic acid. And that will allow for the precipitation of proteins. None of that terminology you'll need to remember, but essentially these techniques employ the precipitation of proteins from other molecules. If fats are present, defatting those tissues with hexane or chloroform methanol mixtures is helpful. After that, purification via solid phase extraction on cartridges ensures for a very clean sample, and that's important. Although the details of the methods are complex, the purity of the sample is key when dissecting the ages present. Recent innovations in these approaches include solid phase microextraction, that's SPME, with covalent organic frameworks for even higher sensitivity and less solvent use. That was seen in analyses of heated chicken and fish employed by this review. Beyond that, bound ages that are bound to big molecules, are even trickier. In this case, they require hydrolysis to release them as free forms. Acid hydrolysis with 6 molar hydrochloric acid at about 110 degrees for 24 hours is common for its speed but also cost effectiveness. Again, your ability to isolate these bound ages in your kitchen is not present. You don't need to do that, but others have in order to fully characterize the age burden in foods. To prevent artifacts, researchers also like to use furescine converting CML. These are samples that are reduced with sodium beforehand. Next, researchers should employ some type of an enzymatic hydrolysis. Sometimes they'll do that with a cocktail of pepsin, pronase E, prolidase, lots of other agents over a period of time. Microwave-assisted acid hydrolysis can shorten it to minutes, boosting efficiency along the way. All of that has been described in this review. But after hydrolysis and the cleanup, Kataoka notes that no one method fits all ages due to stability issues, and performance varies. Recovery rates often hit 80 to 100%, but matrix effects can suppress signals. So for detection, after isolation, researchers employed a technique called liquid chromatography, tandem or at the same time with mass spectrometry. This approach reigns supreme because it is very sensitive, very specific, and it has an ability to handle multiple ages without derivation. So their approaches often employ ultra high performance liquid chromatography. That basically sped things up to where you can isolate ages from a series of different foods in only about 10 minutes. Now, there are some alternatives. For instance, ELISA, enzyme-linked immunosorbent assay, can be a quicker screen, but it's prone to cross-reactivity and interference. So let's move on beyond these isolation techniques, because it is again beyond the scope of the lay people like us that are in the kitchen trying to make choices. But Kataoka basically ends this description by highlighting how these tools have been used to quantify ages across many foods. Meats like excessively roasted pork, they find pack about 40 micrograms of bound CML per gram of tissue. Milk can vary from very trace CML in raw milk to higher milk in processed milk products, or higher ages in processed milk. Breads are a main culprit. Breads can spike during the baking, and the levels in that case are about 140 micrograms of ages per gram of crust material. There are lots of databases, including those that are linked in that publication, that now catalog hundreds of items showing processed animal foods that are grilled excessively and charred being hotspots, sometimes up to 100 times more ages than some of the raw foods. Now, what's the upshot? Well, these analytical advances empower us to minimize dietary age intake. If we opt for steaming over the over-frying approach, if we add antioxidants like maybe green tea, catechins, and other scavenger precursors, or choose low-age staples like fresh, above-ground grown produce, there can be some benefits. But there are some gaps, and the researchers talk about these clearly. The gaps persist. Standardized methods for lab comparability to reduce variability and recovery is an issue. Some of these pre-treatments are automated, and some rely on person-specific techniques. As Kataoka notes, heat-processed foods, especially the overheated, dry-heated, grilled, and charred foods, dominate diets. Refining these techniques might be a key to curbing glycation's toll on your health, and future work should focus on green, high-throughput methods to tackle matrix interference and structural similarities. So why should the everyday person care about these advanced detection techniques or methods? Keep in mind, once again, I'm very thankful you stuck around in this podcast for this long. There's a lot of techniques, a lot of methods, and advanced terminologies that we covered today. Most of that is beyond the scope of where you should focus, in my opinion. Note that there are some advanced detection methods that are used in isolating ages from foods. At the core, these methods provide reliable data that demystifies what's really in our food, going beyond vague nutrition labels to pinpoint hidden risks like high levels of ages. For the common person, this means better informed choices. Imagine checking a public database, if you have access to that, to see that your favorite fast food burger has sky-high ages compared to a home steamed fish fillet. It may prompt you to swap it out a few times a week. Resources like those available from Abertay, the Ages database, have over 8,000 entries on common foods and beverages. And there's a new expanded dietary age database from researchers like Urabari and colleagues. These all make this knowledge more accessible online without needing a science degree. Both are linked at the end of this podcast. By understanding as an overarching principle that dry heat cooking amps up ages while moist methods keep them low, you can tweak daily habits, excessively grill less often, and boil more frequently. Those both approaches can potentially reduce inflammation and lower risks for conditions like diabetes and heart disease over time, we talked about previously. Ultimately, these techniques bridge the gap between lab science and your kitchen, empowering proactive health management through smarter, evidence-based eating. Thank you, everybody, for tuning in today for this slightly more technical version of the College of Glycation. At the end, please remember to keep questioning your plate. Try to understand the age burden that you are partaking, and then we will catch you next time.